I'll start again since Robbie's just turned on the recording. So I wasn't you. Welcome to this August IBH seminar. I'm Tracy Valentine. I'm associated with the IBH as well as the rest of Hutton. And I'm absolutely delighted to introduce and host Gwendolyn Kirschner. Gwendolyn Kirschner recently joined our lab about 9 months ago on a Marie Curie fellowship, post-doctoral fellowship. And she's passed her PhD in Germany originally, working on genetic control of root development. Since she's also worked in Saudi Arabia, and as I said, about 9 months ago, she joined us here in Dundee at the Hutton. So I'll pass over to you now, Gwen, to talk more about your recent research and career up to now on the root angle of barley.
Yeah, thank you. Thank you, Tracy. I've shared my screen. Just a quick confirmation, does it work?
You can.
Looks good.
Perfect. All right, thank you then. Thank you all for coming. Thank you for inviting me. And as Tracy said, I would like to talk about barley and particularly about roots today. First of all, I want to remind you, even though we probably all have it in mind, that climate change will lead to dramatic changes in the climate, obviously, in the future, and will lead to like more extreme weather events. And in Europe, that's already visible. And here you see a graphic of the year 2020, when you can already see first signs of drought all over Europe. And on the right, you see predictions that this might be true for the UK even. Like right now, I cannot really believe it because I have like arrived here 9 months ago, and in my opinion, it has only rained, but I will believe the predictions. And so if we all as lovers of barley want to maintain our barley as healthy as on this picture, we have to eventually adapt it to the new climate conditions, be it drought, flooding, or more infertile soils. And you could have guessed I've started to talk about roots, so I want to draw your attention to this organ of the plant that is usually a bit underexploited, basically because it's not visible by eye, because you have to make major effort to actually see it in the plant. Like if you look at the barley field, you would not at first glance see the roots. But nevertheless, the roots are very important for the plants because they take up the water and nutrients from the soil. And those resources of the soil are not evenly distributed. For example, if we see the distribution of some minerals here, they are plotted in a vertical layers. So the top bit is the upper soil bit, and the lower bit goes down to a meter. And you see that the mineral nutrients are actually sometimes accumulated at the topsoil, like the phosphorus and the potassium in this example. Some are evenly distributed along the whole profile, but some even accumulate in deeper soil layers. This is from a research paper. This is my own data from Scotland this year. And Molly, thanks for the moisture measurements over the summer. And also here you see the soil profile along the depth, basically. And in well-watered conditions where we watered from the top, you see that the water kind of spreads evenly across it. But in drought conditions, where we basically stop watering in spring, you see that the upper soil layers start to dry out. You can also see that lower soil layers still keep a lot of water. So Scotland might actually be, at least in this year, not have been perfect for drought stress experiments. Anyways, what this shows us is that the soil is not even. So we have to kind of look at the root system architecture also in the way that it's important where exactly the roots are located in the soil. And for example, roots that are located at the topsoil have better access to the nutrients that are located there. Like, as I said before, phosphate that would be applied more at the topsoil layer and in the form of fertilizer. While roots that reach deep down into the soil are able to take up deep soil water or mobile nutrients that accumulate there. There are already some examples, some rare examples that manipulating the root architecture actually has an effect on plant performance. So one famous example is the deeper rooting 1 locus in rice, where you can see the wild type deeper rooting 1 on the left, which has a very shallow root system with a lot of roots located at the topsoil or at the top. While in this deeper rooting 1 nearly isogenic line, the root angle is deeper, and thereby more roots are located in deeper soil layers. And indeed, in a drought stress experiment, the researchers could show that the plants with the nearly isogenic line, for deeper rooting 1 performs much better under drought, probably because the plants were able to reach deeper down into the soil to grab the water. So how are those roots actually placed in the soil? A big role for that plays the root angle, because that's the main factor that directs in which bit the roots go into the soil. And what defines the root angle is what is the so-called gravitropic set point angle. That sounds a bit difficult, but basically refers to the angle that is set in relation to gravity and is specific for specific organ type. It also holds true for the shoot, for example, but specific root organ type, specific developmental stage, and specific environmental conditions. And I look at the gravitropic set point angle particularly in barley, because barley has a root system at the seedling that consists of a few different seminal roots, or a few seminal roots that have a different gravitropic set point angle. And the interesting bit is here that they're all the same root type. Seminal roots are all directly derived from the embryo, grow out roughly at the same time, and therefore have the same developmental age and developmental stage. But still, they're able to maintain different gravitropic set point angles. And that is particularly visible in these kind of rotation experiments that are sometimes done when we want to see how a plant reacts to gravity. So what we do for that is we rotate the plant 90 degrees and then follow the root growth over time. And we can see that now the plant is able to maintain the gravitropic set point angles per root. So for example, this root here that has something like a 45-ish degree angle before the rotation just keeps on growing in the same angle because the rotation has not actually affected the angle, and it will just keep on growing in that angle. While the one here on top was located very far from its original angle, and after the rotation now grows back to orient itself back to that angle. And I've already told you that this is a reaction to gravity. So what happens after a rotation in the root tip is that the root is able to sense the gravity in cells that are located at the very tip in the root cap that contain starch granules that are more dense than the cytoplasm and therefore sink down, and thereby trigger a signaling cascade that involves auxin, calcium, and ultimately also pH changes, and ultimately lead to a differential cell elongation in the elongation zone. So in this example here, the root would try to grow downwards towards its original angle again, and thereby elongate the cells at the top of the root much further to kind of push the roots downwards again. But let's go back to the gravitropic set point angle. So the mechanism how plants sense and react to gravity is quite well known, but mostly known in the sense that how they react to rotation. But the gravitropic set point angle is actually an angle that is set constantly. So it's a constant readout of gravity and maintaining this angle. And so far, we don't know how this angle is maintained. And that is what I want to look at in my project. And what I did so far is that I isolated root tips from roots with different angles, and then looked particularly at the tissues that are involved in gravity signaling. So the root cap, the meristem, and the elongation zone. And for that, I used laser capture microdissection, which is a method in which you can isolate tissue, like very specific tissue from samples. And I particularly focused on these zones to eliminate the background that you have if you would just use the whole root, for example, for RNA sequencing. And from that, I would like to be able to correlate the gene expression with angles so that we find out what is the driver that, or what is the internal kind of memory that the roots have to maintain the angle. So that, because there has to be something different between them, otherwise they would not like maintain a different angle and then go back to the different angles. And I would like to first show you a bit more about the method, laser capture microdissection, because I think it's a very important or very interesting technique, but it's a bit widely unknown for some reason. And what you do is you fix your tissue, you embed it in freezing medium, and then you make sections at the cryomicrotome, which then looks something like that. And from that, you're able to go to the laser capture microscope and see this on the screen. And I have a short animation here. And last time I tried it, it worked. So yeah, it needs to work again. And what you do is you tell the microscope just by your mouse where it is supposed to cut in the tissue. And then the microscope activates the laser, a strong laser beam, and cuts through the tissue at the site where you showed it. And with that, you're able to kind of mark the tissue that you want to use. I forgot how long the video is. Great. Yeah. With that, let me go back to the slide before. With that, I was able to isolate the different tissues. So you can basically cut out the root tip, the root cap, and then I isolated the root cap from that, the meristeme, the elongation zone. And I find this method quite interesting because it can even be used for single cell level, but it is much better than just cutting your tissue up, away by a razor blade, for example. And I would like to know, why is it doing it again? A second. Great. I would like to show you some very, very preliminary data that I got, which I still have to discuss in more depth with more experienced bioinformaticians. Yeah, so I took samples, as I said before, of roots with different angles. So a more vertical root, a very vertical root, I mean, a vertical root, one with a shallower angle and one with a very shallow angle. And the most differentially expressed genes I found were actually in the root cap. And you can see that it's at this point not a lot of genes that are differentially expressed, which is also something that I would not actually expect, because the roots they have the same genetic background. I mean, they come from the same plant, they are the same root type, they have the same age, they grow exactly in the same conditions. So you would not expect a lot of changes. So that's something that I have expected. Interestingly, I want to show you the next slide, but my computer is a bit stuck. 1 second. There we go. Interestingly, I found actually differentially expressed genes in a kind of dose-dependent manner. So yeah, I show you here the 3 clusters where I compare the root cap of the different angles. And I could actually find clusters of genes that seem to be dose-dependent depending on which angle they are isolated from. So in the near future, I would like to isolate from this interesting candidate genes, either candidate genes that have maybe a known function in gravity signaling already, but also any other that I can find information of. Analyze their expression pattern in their mutant phenotype to see what the actual role is in setting the angle of the roots. In the very far future, I would also like to integrate proteomics, because at this point we don't actually know if the gravitopic segment angle is regulated on a transcriptional level or on a protein level. It can also be only on a protein level. I mean, probably not only, but additionally on protein level, because a lot of processes in the gravity signaling are organized or are regulated by protein localization or protein phosphorylation. Now I would like to talk to you about a known gene that regulates the root angle that we have found in barley before. And some of you who go regularly to this seminar series will remember Silvio's talk from the beginning of the year, where he already talked in very much detail about how we discovered the gene, because we have worked on it together. So I want to give you a quick summary about that and add a bit more things that I have done, that I have worked on it. So what we found was a mutant from a tilling population that has a very steep root phenotype. So you see on the left the wild type Morex that has a very shallow root angle, and the roots are distributed in a very wide way with a shallow angle. And in our mutant, we found that all roots grow steep down, and that's why the mutant was named enhanced gravitopism 2. Number 2, because there was also a version number 1 of a different gene, number 1, which I'm not going to talk about today. And this phenotype is not only true for the seminal roots in barley, but also for other root types like the crown roots that you can see here from pictures in rhizotrons of older plants, and also for the lateral roots. We were able to find out what is actually causing this phenotype, or we think what is causing the phenotype, and did a similar rotation experiment as we have done, as I've already shown you before. And on top you see the wild type, where the roots adapt after a while again back to their root growth angle. However, in the EGT2 mutant, all the roots react much faster to gravity and then go back to the 90 degrees. So our idea is that EGT2, hence the name, actually reacts stronger to gravity. We were able to map the gene, or to map the mutation to a gene that encodes a sterile alpha motif domain containing protein with a very conserved sterile alpha motif domain. And then we wanted to find out what the actual function of EGT2 is. And for that, we first compared the transcriptome of wild type, of vertically grown roots of wild type, with the vertically grown EGT2 mutant roots. And also here we divided the tissue into root cap, meristem, and elongation zone to be better able to separate between all the processes happening there. And the main thing we found is that in the elongation zone, there were a lot of genes differentially regulated and related to cell wall processes. For example, peroxidases, expansines, and those sugar-related genes with very long names. Additionally, we wanted to understand better what actually happens on a transcriptional level in roots. So we also looked at the, first of all, we looked at the wild type root after a rotation like I've shown you before. So we looked at the transcriptome, again, differentiated between all those zones after the rotation, between the rotation and 12 hours. And interestingly, again, in the elongation zone found a lot of genes differentially regulated. They were related to the cell wall. And in parallel, we did this experiment with EGT2 mutant roots as well, and found a lot of those genes overlapping with the ones that were regulated by EGT2. So our idea is now that EGT2 in wild type case basically blocks the expression of genes that are positively related to gravity, while it rather pushes the expression of genes with a negative role in gravitopism. So in the wild type, we would have the situation that EGT2 kind of acts in the anti-gravitopic pathway, so it maintains the angle, but when we knock it out, the reaction to gravity is enhanced, also based on the transcription. To then better understand the role of EGT2, we looked at protein interactors by yeast to hybrid screen and bifluorescent complementation. Excuse me. Because from the structure of the protein, the role is not really clear, because the sterile alpha domain that is contained in the protein has a lot of functions, but most of it is an interaction domain. So it is known to interact to excuse me, to facilitate the interaction between proteins. And what we found in the screen was two O-methyltransferases that could be again related to cell wall processes, but also a heavy metal transport detoxification superfamily protein with a really long name. And I had a closer look at that. And as I said, we found it in the EGT2 interactor screen, but interestingly, we also found it in a QTL that's related to the seminal root angle that Silvio's group from Bologna kindly shared. And so this makes it a very interesting candidate for being maybe another player in root gravitopism and then root angle regulation. And what I did then was looking at a collection of haplotypes, of different haplotypes of barley, and try to compare the root angle between haplotypes that have the wild type HMT allele with the ones that have a shortened, a truncated version of the HMT gene, and used root angle data from Silvio's group for that. And what I found, and this looks very complicated on the first chance, sorry. I caught a cold. What you can see here is the difference in root angle between one wild type barley line with a haplotype that has a mutated version of the HMT gene, and the difference in root angles. And every dot is basically the comparison, one wild type with a very close related barley allele, barley variety with a mutant allele. And if the dots are above 0, that means that in the mutant version, the root angle is a bit steeper. If it's below, you would say that the root angle is a bit shallower in the mutant. And what we can see is then on average, the difference is above 0. So it seems to be that that a majority of the haplotypes with a truncated version of this HMT gene have a bit of a steeper root angle than the wild type relatives. And what I would like to do next is to actually analyze a mutant of this line, which I would like to do in collaboration with Kelly here at the Hutton. She created a laureate tilling population. And I have the suspicion, or there are indications that this mutant now would have an altered root angle in comparison to wild type. And it doesn't seem to have a root angle phenotype that's as strong as the EGT2 mutant, but only a very slightly altered angle, which would be very interesting to compare then in terms of how does it perform in soil, how does it perform in different environments. And I would also like to go on and analyze the connection of the root angle to heavy metals, because there are first indications that the occurrence of heavy metals in the soil alters the root angle, which would also make sense because heavy metals are also not evenly distributed in the soil, and maybe there is a mechanism of plants that are actually making the plant avoid the patches or the layers with heavy metals. And by analyzing both the regulators of the gravitopic set point angle as well as the EGT2 signaling network further, I hope that I can better understand root angular relation and then ultimately be able to adapt the root system angle or the angle of single root types so that we create plants that better perform under specific environmental conditions. With this, I would like to thank everyone who has so far been involved in the EGT2 story. mainly in Frank's lab, Silvio's lab, also another lab here in the UK, the John Center. The IBH was involved as well, Gießen was involved as well, and a different lab in Bonn. I would like to thank Silvio's group in Bologna for providing all the data for the root angle, in which I used to analyze the HMT gene. Collaborators in Marburg, a lot of people here at the Hutton, and I'm sure I forgot some to list some of them. Thank you. And my funding. And yeah, I would be happy now to take questions.
Thanks very much, Gwen. Take a couple of seconds just to take a drink if you like. Does anyone
Yeah, sorry.
have questions
Oh my God.
for Gwen? Well, I'll start off one. Oh, Robbie, you go first.
Gwen, what's the extent of the natural variation for set point angle in the roots?
Oh, I'm sorry, can you repeat? I was coughing mentally.
What's the extent, sorry, it's my accent. What's the extent? Germans find it particularly difficult to understand. Yeah, what's the extent of natural variation in set point angle in barley roots? Have you any idea?
In barley, I cannot remember any publication about that. So I'm not sure if I'm not aware of it or it hasn't been done. I mean, let me think. No, there is a variation actually, because Silvio has this looked at the root angle of the Welby population. So
Okay.
there is a variation, but it has not yet been mapped to specific geographical locations or to specific, it hasn't been related to any plant performances yet.
So I should probably give you the context of the question, and
Maybe.
it was because I listened to a talk last week by Lee Hickey from Queensland.
Yes, it must be wheat.
And Lee, no, it was in barley actually.
Oh, really? Yeah.
But Lee did some experiments looking at root angle on a panel of 384 accessions from an Australian gene bank panel. And if I remember correctly, he came to the conclusion that set point angle or root angle was controlled by lots and lots of genes, minor genes. He said there was no major genes involved, which of course immediately made me think of EGT2 and 1. And I thought what he was talking was just a little bit uninformed. But he said basically it would take variation in 3,000 minor genes to alter root set point angle by 30 degrees. Does that make sense?
For me, that actually makes sense, because I think EGT1 and EGT2 are not necessarily regulators of the gravitopic set point angle. They are regulators of gravitopism. And if you knock them out, you just knock out. I mean, you just kind of knock out the whole pathway that would make a more shallow angle. So
okay.
I would rather, like, we still don't know if maybe dose-dependent expression of that would change the root angle, but if you knock it out, you just switch off that whole pathway, so you make it a steep root. You don't vary the root angle, you just make steep roots, because they can only do one thing and that's growing downwards. They lost the completely whole pathway for the anti-gravitophic bit.
Okay, so
that's actually possible, yeah.
So
but I also remember that Silvio actually found only one QTL for his root angle.
Yeah.
But I would have to come back to the seminar from this beginning of this year again.
Yeah, I mean, to be fair, Lee was talking about this in terms of using genomic selection to alter root point angle. And he reckoned there were like 3,000 or so factors involved in controlling this to actually change the angle by 30 degrees, it would be, wouldn't it, 30 degrees. So maybe I just misinterpreted what he was saying, because I thought 3,000 genes seems a bit strange when we know that if you knock out EGT2, then you can probably change the root angle quite significantly with one gene. But anyway, I think you've probably explained it for me, which is good. Thank you.
I suppose one of the difficulties is the difference between something that's controlling it in that dose response versus all the cascade of everything that goes into changing that angle.
Sure.
A question from Laurie.
It's not Laurie, it's Tim, but that's fine. So Gwen, the sensing of gravity is a physical process. So as you say, the statoliths or whatever, these starch granules accumulate in the cells and it sends a signal. Do you know what would happen if you grew plants in zero gravity? So if you were able to go up into the space station and grow some plants, what would happen to the root growth?
I feel that I should know because I think it has been done, but I don't remember. Like I think someone took either beans or Arabidopsis to space already. Yeah, I don't remember what the roots do.
Would they just go everywhere, do you think? Or
Like what I would think, just from my knowledge, that they just go at the next parameter that they are interested in, because there are also things like heterotropism and so on, or like the growth away from light, I don't know, towards nutrients. So I think they would just go to the next level of control.
cool, thanks.
But there are studies about it. I'm just, don't have the results in mind.
Yeah, it's really interesting. I was just wondering, that's possibly a naive question. I was wondering whether you looked at the mechanical properties of the tissue itself. The easy way to describe it is like cooked spaghetti and raw spaghetti, whether there's any kind of inherent stiffness or mechanical property of the root related to the genes that you're looking at. And I was wondering if that could explain why if it's much more flexible, it would respond quicker to changes in orientation that you showed in your images.
Yeah, so we have not done it for EGT2. The group in Nottingham has done it for EGT1, which ended up having a bit of the same or similar differentially expressed genes. And they actually used atomic force microscopy to look at the cell wall, I think in the meristeme or elongation zone, and found that there's a difference to wild type. So it turned out, now I have to think, that in the wild type, the cell wall in the cortex, I think, is a bit stiffer. So their hypothesis is actually that EGT1 kind of regulates the stiffness of the cell wall, and that thereby have these kind of anti-gravitropic counter force to not make the roots grow downward.
Okay, and then
not true for EGT2.
Okay, and then a follow-up question to that then. So has there been any studies looking at the relationship to anti-gravitropism and mechanical impedance to root elongation? So I wonder whether if you have this reduction in cell wall mechanical properties, whether it influences like rooting depth or something like that under soils of differing bulk density. I wonder if that's a quick screen potentially as well.
I'm not aware of any study that is related to soil density at all. I mean, I only know for our mutant, for our EGT2, that the roots grow the same speed, but only under controlled conditions. Like also in soil and rhizotrons, but they are also not particularly dense. So I don't know under extreme conditions, but I think it would be interesting actually, yeah.
Yeah, we could maybe have a chat about it. Could it be interesting? You could also do a screen under a structured soil and an unstructured soils, based on the assumption that the ones that anti-gravitropism can influence where the roots can explore the soils. But yeah, can chat about it later.
Yeah, absolutely. Would be great.
Okay, thanks, Kath. Anybody else? Well,
yeah, nice presentation. Thank you. I have a question because I remember quite some studies from the past in Arabidopsis roots where you looked at this gravity and the bending and the oxen gradients. And I was wondering to what extent this type of studies are useful for you. Can we translate it to barley, or do you have to start all over again? Because I saw you did the RNA-seq in the bending as well. So how does this overlap, this type of studies with yours?
Yeah, so what we know so far for all the bending experiments Of course, there is much less known in general in barley. We don't have a auxin reporter line and so on. But so far, from the mechanics, everything seems to overlap with Arabidopsis. The only thing that we cannot really transfer is the thing about the gravitopic set point angle, because of course, barley has a completely different root system architecture. So in Arabidopsis, you have this primary root that basically only does vertical. So you cannot really do any studies about the angle. You can also not identify any mutants based on that because the set point is already down. And that's
Exactly, because that's why I'm asking, because the architecture from the Arabidopsis roots are really different when compared to the cereals, right? So
yeah.
Yeah.
Yeah, so it's more, that's why I'm actually also using barley, because in the root system, there are different set point angles. So we can transfer from Arabidopsis the reaction to the rotation, I would say, but we cannot use Arabidopsis really for the gravitopic set point angle, because there is only this one vertical one in the primary root. And the lateral roots of Arabidopsis, of course, don't have a vertical set point, but a bit more shallow. But there, it's also difficult to use them for studies because there all the lateral roots have basically a similar angle across the plant.
Yeah.
So, yeah.
So basically you have an important message, right, in that sense. So if you really want to understand what the roots, like how the architecture is arranged in crops, you have to study it in the crop itself.
Yeah, I think so, yeah.
Yeah. Okay, yeah, thanks for clarifying. Thanks.
And question from James Reeves.
Yes, this honestly, I have a feeling I know the answer now because of what you just said about Arabidopsis, but just in case, do you have any idea how well conserved these genes are across cereals?
Yeah, so so far for EGT1, not 100% sure, but for EGT2 for sure, we have mutants in wheat, and they show a very similar phenotype. So if you knock out both homologs of EGT2 in wheat, they also show this downward growth of the roots. EGT2 is even like conserved across dicot plants, so you also have a conserved function in Arabidopsis, where it was more difficult to find because I was already saying that the primary root in Arabidopsis is already growing downwards. So in a screen, you would never really find it if you don't pay attention. And the phenotype in Arabidopsis is that lateral roots now grow in a steeper angle. And EGT2 even has a function for gravity signaling in peach, where it was first discovered for the branches. And if you knock it out, you have like I think it's a weeping phenotype where you have like a steeper angle of the peach branches. I'm not sure if that's the right wording for peach trees. But yeah, so it seems to be a quite conserved gene among all kind of plants.
So anyone? One more from Robbie then.
Just a quick one. I'm interested in the HMT proteins.
Mm-hmm.
Do either the EGT2 mutants or any of the HMT haplotypes respond differently to, for example, the presence of cadmium or lead or arsenate in the growth medium?
Yeah, that I don't know yet. And it's also not, that's something I would like to start to check, but I haven't found anything about root angle changes, particularly in barley. I think there's one paper in Arabidopsis, but nothing apart from that. So I don't even know if they changed their root angle. It's just an educated guess.
And sorry, same line. Is HMT something to do with siderophores or not?
Not by putting it into the general kind of protein structure databases. So it only ever comes up as, I think, copper transporter or something.
Ah, interesting. I think Frank's trying to get in there with a little comment. I see his screen's flashing on and off. So I'll be quiet now.
Frank, were you trying to say something?
Hi, can you hear me?
Yes.
I was wondering about that LCM RNA-seq study that you showed in the beginning. You found a relatively small number of differentially expressed genes. So I was wondering the total number of expressed genes that you had in your RNA-seq data. How high was that, or how many active genes did you find in that approximately?
Oh no, I don't remember now. I have to look it up. I don't remember. The numbers are
Okay,
a
no
bit
problem.
surreal for me, so that's
Yeah.
yeah, I didn't save them in my head.
I was just wondering if you found thousands of active genes and only maybe 30 or 40 differentially expressed ones, or if the the number of expressed genes was also relatively low because of your LCM procedure.
Yeah, no, I have to double check that. Thanks.
Okay,
Yeah.
no problem.
So, any more questions? Answer. So I was just going to ask one more myself then, Gwen. The mutants you talked about, they all seem to have enhanced gravitropism. Do you have any germplasm where you've got roots that are going almost horizontal, and that's their set point angle? So almost the opposite of your knockout mutants.
Yeah, no. I haven't heard of anyone who found anything with a more shallow root angle in comparison to wild type yet. Yeah, so no.
Right. So if there's no more questions, I'd like to bring this to a close, and I'd like everybody to thank for a fantastic talk. Very much.
Thank you very much. Thank you very much for listening.
Thank you.
Thanks, Gwen. Well done.